बहुत बत्ती मुंह पे लग रही है, क्या लगा दिया है? Oh, shit.
कर दो स्टार्ट, कर दो आप स्टार्ट।
Good afternoon, everyone. A very warm welcome to another live session for AIATS Plus Phase 2, where we discuss the questions and concepts in which most of you tend to make mistakes. So far, my colleagues have covered many topics concerning to zoology, but yet there are some important topics left which should be discussed when it comes to the NEET exam. And I am Snehil, your zoology faculty. So, the topics that I am going to cover in this session are biomolecules, excretory product and their elimination, locomotion and movement, human health and disease, and evolution. And like I always mention that amongst all, it is biomolecules from which most of the questions are asked in the NEET exam. Alright, so let's begin the discussion without wasting much time. So the first question that we have today in our bucket is question number 136, and it pertains to the chapter biomolecule, as I can see, and it states cytidine is a nucleoside of RNA. It has. Alright, I can see students have joined. So it's good afternoon and not good morning. So a very warm good afternoon to everyone. I hope you enjoy the session. So by looking at the question, I can see that it is talking about the types of bond which constitute the structure of a nucleoside. So first of all, let us discuss that where is this nucleoside coming from. So we all know that the basic genetic material of humans is DNA, and DNA and RNA are said to be the genetic material. Alright, they are the nucleic acids which act as the genetic material. And our NCERT states that nucleic acids are composed of three basic structures, and they are a phosphate group and a monosaccharide, and a monosaccharide can be a ribose sugar or deoxyribose, depending on the type of genetic material that we are talking about. I hope you are aware of the fact that there is a structural difference between ribose and deoxyribose, and in DNA deoxyribose is found, as the name suggests itself. whereas in RNA, ribose is present. And what is that structural difference? I would like to know from you in the comment section. All right. Again, the last component that we have in the structure of nucleic acids is the nitrogenous bases. So the nitrogenous bases are the component of the nucleoside that we were talking about in our given question. Now let us see what are these different type of nitrogenous bases which constitute the structure of nucleic acids. Also, it is important to remember that the monosaccharide that we are talking about here is a ribose sugar or a hexose sugar. Please let me know in the comment section. So basically, the sugar which is present in nucleic acids is a pentose sugar. All right, so coming back to our nitrogenous bases, the two type of nitrogenous bases that we talk about are purines and pyrimidines. Now, purines are the nitrogenous bases which constitute or which are made up of two heterocyclic rings. All right. So basically, what we mean by a heterocyclic ring is that we will have a structure, as you can see in the picture, we will have a structure which will be made up of atoms which are different from the carbon atom. So basically, in a simpler way, we can say that a heterocyclic ring means a ring in which there are atoms present apart from the carbon atom. So here we have the nitrogen atom, and that is why we are calling it a heterocyclic ring. So often this question is also asked in the exam that which molecules contain heterocyclic rings, and the answer is our nitrogen bases, also our ribose sugar, glucose, because in all these structures there are atoms present which are different from the carbon atom. All right. So in purines we have two examples, and they are adenine and guanine. All right. I can see students have answered ribose sugar and deoxyribose, so you are absolutely right. Okay. So on your left hand that you can see is the structure of adenosine. We will discuss where this adenosine is coming from, but you should know that this whole part of the molecule represents adenine. All right. This is my adenine. Similarly, the molecule shown here represents guanosine, and the circle part represents the guanine molecule. Similarly, if we talk about the pyrimidines, so the pyrimidines are characterized by the presence of only a single heterocyclic ring. As you can see in the figure, we have our pyrimidines, and we can see that there is only one ring present in the structure. And the examples of pyrimidines include thymine and cytosine. Whereas the basic structural difference between RNA and DNA is, first of all, the presence of different types of sugar, and the other difference that comes into picture is the presence of uracil in RNA instead of thymine. All right. So this was the basic concept pertaining to nitrogenous bases and the different components that constitute the structure of nucleic acids. But our basic question was about nucleoside, all right? So now going back to that, so basically let's break the structure of nucleic acids into smaller fragments, all right? So basically if we take DNA, then it is said that nucleotides are the building blocks of nucleic acids. So basically many nucleotides will come together, or we can say that DNA is a polymer of different nucleotides. Again our question said nucleoside. तो ये side and tide में difference क्या है? That is what we want to decipher, right? So here I am saying that it is made up of nucleotides. अभी nucleotide को अगर हम further break करेंगे, then we will have nitrogen bases plus our sugar, and the sugars can be ribose and glucose as I explained before, plus the other component is the phosphate group, all right. So basically the nitrogen base and sugar together, they are said to be nucleoside. So it is important that you understand the difference between nucleoside and nucleotide, all right? So these two constitute the nucleoside, whereas if we add phosphate group to it, then together it will be known as nucleotide. All right. Now if these molecules are being joined, then of course there will be some sort of bond present between the two molecules to join them together. Okay, all right, I can see people asking to use the yellow color. I'll use that. Thank you for pointing out. So okay, so the bond which is present between the nitrogen base and sugar is the glycosidic bond. All right, and the bond which is present between the sugar and the phosphate is the ester bond. All right, and in the picture shown here, what you see is a structure of a nucleotide or a nucleoside. Let me know in the comment section. So basically it is representing a structure of a nucleotide, and we can see that our nitrogen base is connected to the pentose sugar, and this bond will be known as the glycosidic bond, whereas the OH group of the sugar and the O group of the phosphate group will together form the ester bond. So here that you see is the ester bond, all right. So basically these bonds are coming together to form the structure of nucleotide, and then the polymer of nucleotide will together form the structure of the DNA that you see on the screen. So this is just one strand of the DNA, all right, and you can see the ester bonds that we have, and the ester bonds are specifically formed by the OH group which is present on the 3' carbon, and further it will bind with the O group which is present on the 5' carbon of the succeeding nucleotide, all right. In the picture it is not very clearly shown, but it is always the the bond is always formed between the succeeding nucleotides. All right. So coming back to our question now, we had cytidine. So cytidine here is an nitrogen base. So here it is a nitrogen base. All right. Sorry, cytidine. Cytosine is my nitrogen base, not cytidine. So basically my cytosine was the nitrogen base, and when I added sugar to it, it became a nucleoside, and it will be known as cytidine. And further, if I add phosphate group to it, it will be known as the cytidyllic acid, and this is my nucleotide. And now let us read the options one by one to see whether the description given about the bonds is correct or not. So our option 1 states that ester bond between ribose sugar and phosphate group. So as we have discussed, that since it is a nucleoside, there will be no phosphate group. Therefore, our option 1 is incorrect and cannot be the right answer. Moving to option 2, it states glycosidic bond between the ribose sugar and cytosine base. So our option 2 seems to be the right answer. But let us read the other options as well. So our option 3 states hydrogen bond between ribose sugar and phosphate group. So again, we don't have a phosphate group, therefore option 3 cannot be the right answer. Again, in option 4 I can see ether bond and phosphate group. So ether bond is not present, so it is an irrelevant option altogether. So the basic takeaway message here is that glycosidic bond and ester bond constitute the structure of nucleotide, and we need to remember the groups between which these bonds are specifically present. All right. So now we move on to our next question. All right. So our next question is question 138. And I can see in the comment section students have answered it correctly, that the answer is option 2, and you are right. All right. So now moving to question 138, and it states all of the following statements are correct regarding enzymes except, and I can see big statements. All right. And I can see the keywords that we have: coenzymes, competitive inhibition, Vmax, 10-degree change and whatnot. So let us quickly revise these concepts so that we can figure out our right answer. All right. So first of all, let us begin with the basic one. When we talk about the cofactors that are involved in enzymes. So this topic has already been dealt in previous classes in great detail. So here I'll just give a quick recap. All right. So basically there are 3 types of cofactors that are present. Basically our enzymes are the protein part, and it needs a non-protein part which is known as the cofactor to make this enzyme catalytically active. It is very important that we focus on the words catalytic activity should be present in an enzyme. Only then it will be known as the holoenzyme. Whereas if this non-protein part is not present, then this enzyme is still a protein, but this will become catalytically inactive, and that protein will be known as the apoenzyme. So it is very important to remember that for an enzyme to function properly or to show catalytic activity, cofactors should be present. So let us quickly revise the factors that we have already discussed before. So we have prosthetic groups, then we have the coenzymes. I would like to know the examples of these from you in the comment section, since we have already discussed it before. And then finally we have the metal ions. I'm waiting for your answers. All right. So the prosthetic group that we talk about is organic in nature. It is important that we focus on the nature of these cofactors, because in NEET exam questions are often asked whether they are organic in nature or inorganic in nature. All right. So prosthetic groups are organic in nature. They bind tightly to the apoenzyme, or we can also call it, they form a permanent bond. All right. Then moving to coenzyme. In coenzyme, we have two examples which is given in the NCERT, and they are NADH, and NADH is said to contain the vitamin niacin, and this is again asked in exams, that whether a vitamin component is present in coenzymes or not. So the answer will be yes, a vitamin component is often present in the coenzyme. So again, if we talk about the nature of coenzymes, then they are also organic in nature, but a remarkable difference between the prosthetic group and coenzyme is that the coenzymes do not bind to the apoenzyme tightly, or we can also say that the bond formed between the coenzymes and the apoenzyme are said to be transient, or they bind loosely with the apoenzyme. All right. Coming to metal ions. So metal ions are again can be of we can make out that the nature of metal ions is obviously inorganic here, and the example that is mentioned in our NCERT is zinc. So zinc acts as a cofactor for an enzyme which is known as the carboxypeptidase. All right. So this is the basic concept of cofactors, and exactly these specific points are given in the NCERT, which we have covered very quickly. All right. So now we move on to our next concept, where we talk about the factors that bring about a change in the rate of reaction. So what exactly is, I can see someone has commented that prosthetic enzyme FAD. So you're right, FAD is also one of the cofactors. All right. Okay. So basically when we talk about rate, so rate means the velocity or the speed with which a reaction is occurring. All right. So there are various factors that can influence the rate of reaction. Okay. And these factors are temperature, pH, presence of inhibitors, and finally we have the concentration of substrate. So any change in these factors will bring about a change in the concentration, in the rate of the reaction, meaning it will bring about a change in the speed or velocity with which the reaction is occurring. So basically when we talk about temperature and pH, then there are optimal values at which reactions occur when we talk in terms of pH value or temperature. So if there is any change beyond this optimal value, be it any increase or decrease, it will affect the rate of reaction. it can be mostly it is a negative effect, which means that the rate of reaction will decrease because these change in these factors brings about a change in the structure of the enzyme which is made up of proteins. So any change in these factors disrupt the protein structure or the bonds which hold that structure together. All right. Now coming to the other factors, which is the presence of inhibitors that we will discuss in greater detail. All right. And when we talk about the concentration of substrate, that will be also discussed. So coming to temperature now, I can see that a snippet has been placed here, and you can also see on your screens. This line is basically given in the NCERT, and it simply states that rates of reaction of physical and chemical process are influenced by temperature. So it is true, we already discussed that. Among other factors, a general rule of thumb is that rate doubles or decreases by half for every 10-degree change in either direction. All right. So basically, if we consider that we have two substrates, for instance, and they are combining together to make a product, we always know that reaction can be in both the directions. All right. So here either direction means exactly the same, that if there is any 10-degree change, which means if we increase the temperature by 10 degree, our rate of reaction will double, and it will favor the forward movement of, or it will favor the conversion of substrate into products. All right. Whereas if we decrease the temperature by 10 degree Celsius, it will reduce the reaction by half. Reduce the rate of reaction by half, which means our reaction will be now running in the backward direction. So basically, change in temperature can bring about a change in the rate of reaction, and for that we have mathematical values, which means if the reaction rate will increase by double if we increase the temperature, and the reaction rate will become half if we decrease the temperature by 10 degree. So if there is any question asked where you have been said that if there is any change in temperature by 20 degree, so we will apply simple maths here, and we will calculate the factor by which the reaction rate should increase, and it will be 4 times. So it's just basic maths that you need to apply, and we might get such questions, calculative questions, where we have been asked that a certain temperature is given, and if there is any change in the temperature values, what will be the rate of reaction. So we just need to apply the basic maths here, that will double the rate of reaction if it is an increase in temperature, and will reduce the rate of reaction by half if it is a decrease in temperature. All right. So these factors, so we have covered temperature now. Coming to pH, I already explained that change in pH will disrupt the structure of protein, and therefore the activity of enzymes gets disturbed. All right. Now that you see on the screen is a graph which is taken from the NCERT, and I can see that there are various values, I would say, written here, जिसका NCERT में कोई explanation नहीं है. So we will try to figure out what exactly these symbols mean. Okay. So if we talk about Vmax first, so Vmax is nothing. It is simply the velocity of reaction when it is at the maximum value. which means that the enzyme can reach this Vmax value only. इससे ज़्यादा the rate of reaction is not going to increase. It is already functioning at their maximum capacity, and that is exactly what has been shown in the figure also, that the rate of reaction has reached its maximum value. All right. So Vmax is simply the rate of reaction at the maximum value. All right. Coming to the other terminology or the symbol, which is Vmax by 2. So this also simply means that the value given here will represent half the number of the maximum value, which means जो maximum value यहाँ पे हमें attain होगी उसका half कितना है, that is what is represented by Vmax by 2. It is simply, it is very easy to make out because we can see the mathematical here formula here, that it is just half the Vmax. All right. So basically it is half the value of Vmax. All right. Now coming to Km, which is the one of the important values. So Km basically means that it is, Km indicates the concentration of substrate So Km represents the concentration of substrate at which my reaction will reach the Vmax value. So if asked in the question what exactly is Km, so Km is that concentration of substrate where the reaction reaches Vmax, and this Km is also known as the Michaelis-Menten constant. It is known as the Michaelis-Menten constant. So basically Km is used to study the affinity of enzyme for that particular substrate. All right. So we can make out the affinity of enzyme for the given substrate. To put it simply, just imagine that for a reaction, my Km value is lying somewhere here. If my Km value lies somewhere here, it means that I need more amount of substrate to reach this Vmax value. All right. So this will simply mean that the affinity of enzyme for that substrate is low, क्योंकि हमें ज़्यादा substrate लगाना पड़ रहा है Vmax को reach करने के लिए. All right. So Km can be used to test the affinity. यहाँ पे if we compare the two, 1 and 2, यहाँ पे हमें कम substrate चाहिए to reach Vmax by 2, which means यहाँ हमें जो हमारा enzyme है वो बहुत readily substrate से bind कर रहा है and they are reaching the Vmax value very quickly. All right. So basically, to determine the functionality of enzyme, Km values can be used. So it acts as a predictor whether the enzyme is functioning properly or not. Okay. Now coming to our inhibitors and how they influence the reaction is, let us discuss our inhibitors now. So inhibitors are nothing but basically such molecules that disturb the enzyme substrate interaction and lead to the disturbance in the rate of reaction. So what are the two types of inhibition that you have heard? Please let me know in the comment section. So our NCERT talks about competitive inhibition only, whereas we have two types of inhibitions. One is the competitive inhibition and the other is non-competitive inhibition. All right. So basically, the prominent examples that we are going to study about when it comes to competitive inhibition is the succinate dehydrogenase, the inhibition of succinate dehydrogenase by malonate and oxaloacetate. And the other example, which is used at the economical scale, I would say, is the killing of bacterial colonies. And both these examples are given in our NCERT. Okay, so let us first of all discuss the competitive inhibition. So you can see in the diagram that it is trying to display how the competitive inhibition works. So basically what happens is, when we talk about competitive inhibition, we have our enzyme, and enzymes have particular sites which are known as the active sites where the substrate binds. These active sites are also known as the pockets or crevices, all right. And we have our substrate here as well. So when we talk about competitive inhibition, what happens is that the structural similarity between the inhibitor and substrate is at a greater degree, which means सबस्ट्रेट और इनहिबिटर एक दूसरे को इतना ज़्यादा रिजेंबल करता है कि both of them have a competition to bind to the given active site, and that is why it is being said competitive inhibitor, because both of them resemble their structure and then they are trying to bind to the active site of the enzyme, and this is how they hamper the activity of the enzyme. They will bind to the active site. The inhibitor will bind to the active site of the enzyme and it will prevent the substrate from binding. तो हमारा जो major goal था to produce a product from a particular substrate will go haywire, alright? हो ही नहीं पाएगा वो, ठीक है? So we can see in the picture also that our active site has been taken over by the inhibitor and not the substrate. And why did it happen? Because there is structural similarity between the substrate molecule and the inhibitor molecule. Alright, we can see that the substrate, the poor substrate is hanging outside. It could not get into the active site, alright. So we have our enzyme inhibitor complex formed instead of the enzyme substrate complex, alright. And the examples that are given in the NCERT talks about the succinic dehydrogenase, so basically what happens is that succinic dehydrogenase is our enzyme, and to bind to it there is a substrate which is known as the succinate. So if we draw the structure of succinate, it is COO, CH2, CH2 and COO. And if we draw the structure of malonate, it is somewhat similar. We will see that. So you can see that there is a high degree of similarity between the structure of succinate and malonate. So basically what happens is that the malonate will compete with succinate to bind with the malonate will compete with the succinate to bind with the enzyme which is the succinate dehydrogenase. So these two are going to compete with each other, and eventually malonate will bind to our enzyme, and thus it will inhibit the activity of our enzyme, and this is what is known as the competitive inhibition, because these two molecules are competing amongst each other to bind to succinic dehydrogenase. So now here comes the role of concentration of substrate, which we talked about, that how it is going to affect our enzyme activity. So basically what happens that this binding of inhibitor with the enzyme is a reversible binding. It is a reversible binding. So if we increase the concentration of our substrate, concentration of substrate is increased, then the enzyme inhibitor complex formed will get diluted, and it gets diluted to an extent that the substrate now will take over and it will start binding to the active site of the enzyme. So basically by increasing the concentration of substrate, we overcame the inhibition which was existing, and that is why we consider that the concentration of substrate also plays a major role when we talk about the factors that affect the enzyme activity. All right. I can see that students are asking in the question that whether I am discussing the concept or the question. So dear student, it is both. Basically we are trying to cover the concept which is behind every question where you tend to make mistake, so that you don't make the same mistake again. All right. Because question ka answer to ऐसे भी we get to know somehow, but what is the underlying concept and how you should approach the question, that matters. Okay, coming to our another category, which is the non-competitive inhibitor. All right. So basically what happens here is, why we are calling it as non-competitive and not, there is a difference in the name. So where is that difference coming from? So basically here what happens is that we can see we have our substrate and we have our inhibitor. All right. So here we can see that the shape of both of them are very different from each other. तो ऐसा कुछ है ही नहीं कि दोनों के बीच में सिमिलैरिटी है, तो कंपटीशन होगा ही क्यों? So basically in non-competitive inhibitor, what happens is we have one inhibitor and it will bind to one of the active sites of the enzyme, and it is so evil that it will change the shape of the active site where the substrate was supposed to bind. All right. We can see in the figure, like in the first figure, we can make out that the shape of the active site is complementary to the shape of the substrate. But when this evil got attached to the enzyme, it changed the shape of the active site. It became broader. All right. So even if our enzyme will, our substrate will try to fit in, it will not, and therefore that is how this reaction will now get, the rate of this reaction will now get inhibited. All right. And therefore, we can see it also states change the shape of the active site, and therefore, there will be no formation of enzyme substrate complex, and this inhibition cannot be overcome by increasing the concentration of substrate, and therefore, we also call it as the irreversible inhibition. All right. So now we go back to our question. And let us read the statement one by one to find out our answer. So our option 1 states control of bacterial pathogen using sulfa drugs is an example of competitive inhibition. So we already discussed that under competitive inhibition we have this example where we kill the bacterial colonies. So what exactly happens here is that in our body or in general across animals, there is one acid which is required for normal cell growth and development, and that is the folic acid. So when we talk about the bacteria have a cell wall, we are aware of that. So the bacteria cannot take this folic acid from outside, and therefore it tries to synthesize this folic acid for their growth by the precursor molecules that is present in their body, and that precursor molecule is PABA, which is para amino benzoic acid. So if I draw the structure of PABA, it is somewhat like this. We have a benzene ring. All right. So what happens is in experiments or at a commercial scale, sulfa drugs is used that will compete with PABA to bind to the site from where folic acid was supposed to be synthesized. Okay. So the structure of sulfa drug is somewhat like this. We have a benzene ring, then the NH2, sulfa drug is somewhat like this. All right. So basically we can see that there is a structural similarity between the sulfa drug and the PABA, and therefore this sulfa drug is going to bind to the active site or the enzyme from where folic acid was supposed to be synthesized. So folic acid synthesized ही नहीं होगा फिर bacteria में. So normal cell growth will not be seen in bacteria, and therefore the colonies will start dying due to lack of folic acid. And this is how control of bacterial pathogens is performed by using sulfa drugs, and it is illustrating an example of competitive inhibition because PABA and sulfa drugs have a structural similarity. All right. Therefore, option 1 is correct and cannot be a right answer. Moving to option 2, which states coenzymes are organic compounds, which is true as we discussed, and they are tightly bound to holoenzyme throughout the reaction. So we discussed this in great detail that they are not tightly bound, instead they are loosely bound, and therefore our right answer seems to be option 2 itself, because we are looking for the incorrect statement. All of them are said to be correct. But let us read the other options as well, where our third option states once Vmax is achieved, no further change in reaction velocity occurs due to lack of free enzyme molecules. So basically what happens is that if you see the graph that we had here, if you see the graph, what happens is as we keep on increasing the substrate, and as the substrate is increasing, our rate of reaction is also increasing, but there comes a point from where it will start going to infinity. There will be no further rise in the Vmax value. And why does this happen? It happens because enzymes act through active sites. All right. So even if, so what happens is the substrate is going to bind to these active sites. So there comes a point when all the active sites present in the enzyme gets saturated, and then even if we keep adding on more substrate, कोई point ही नहीं है, because सारे यहाँ पे जो मेरे active sites हैं they are already occupied. So अगर हम substrate add कर भी रहे हैं तो where will it bind? There is no place to bind. Therefore further increase in Vmax will not be observed. So basically the saturation of active sites lead to this, that there will be no more increase in Vmax value. Therefore our answer option third cannot be the right answer. Moving to the last question, which states rate of reaction doubles or decreases by half for every 10-degree change in temperature in either direction, and we discussed already this is absolutely true. Therefore option fourth also cannot be the right answer, and thus a right answer is option 2 itself. Now moving on to our next question. So our next question, let's see, pertains to lecithin. Okay, so again a concept question of biomolecules. So we can figure out how important is this chapter, biomolecules, because we have very tricky questions and lengthy questions from this chapter. All right. So among the options given, we need to choose the incorrect one when it comes to lecithin. So what exactly is lecithin? So we can see the structure of lecithin on our screen. So lecithin is basically a lipid, and to be precise, it is a phospholipid. And how are we calling it as a phospholipid? We can see in our diagram itself that we have a phosphate molecule in the structure. All right. So basically lecithin is a phospholipid which is predominantly found in the cell membrane, and we can say that it is made up of two fatty acids. The two fatty acids that we have here are R1 and R2. All right. So it is composed of two fatty acids and we can also see that there is another molecule present in the structure which contains the nitrogen atoms, and this is known as the choline molecule. And this question is often asked that which molecules are present in the structure of lecithin, does it have any nitrogen atom or not? So students often tend to miss out that there is a nitrogen atom which is present in the structure of lecithin, and it is known as the choline molecule. Okay, all right. And as we all know that lipids are also formed by, it is not a polymer, but it is formed by dehydration synthesis, and there will be ester bond present in the structure of lecithin as well. So let us quickly go back to our question to find out our answer, and our option 1 states found in cell membrane, which is absolutely true and cannot be the right answer. Option 2 states possess 3 fatty acids. So this is incorrect, as we discussed and saw in the figure also that it has 2 fatty acids and not 3 fatty acids. Therefore our right answer will be option 2. But let us see the other options as well, where option 3 states it is a phosphorylated organic compound, which is absolutely true. We saw the presence of a phosphate group in the structure. Therefore option 3 also cannot be the right answer. And lastly we have contains a nitrogen atom in its structure, which was the choline group that we are talking about here. So this is also correct and cannot be the right answer. So our right answer is option 2. So dear students, this marks the end of the discussion for the zoology portion. I hope you enjoyed the session, but stay tuned because we have our esteemed botany faculties joining you soon for the discussion of the questions pertaining to the botany section. And all the best for your upcoming NEET exam. The dates are out as well. All the best. Take care and hope to see you soon. Thank you.